A public service announcement regarding stem cell research and gene patenting. Sources are listed throughout the presentation. 1 star is a source, 2 is from the National Science Digital Library, and 3 is from an open access journal. Stem cells. Stem cells are undifferentiated cells, meaning they have no determined function or structure. They have 2 main characteristics. 1, they are capable of renewing themselves through mitosis for long periods of time and sometimes after long periods of inactivity. And 2, they can be chemically induced into organ-specific cells. Now onto the proponents of stem cell research. The primary goal of stem cell work is to identify how undifferentiated cells become the cells that form tissue and organs. A more complete understanding of that genetic and molecular control will yield information about diseases that arise and suggest new strategies for therapy. Additionally, stem cells are currently being used to test new drugs. For example, cancer cell lines are used to screen potential anti-tumor drugs. The most famous cancer cell line, HeLa, named after the woman whose tumor the cells came from, was grown by Dr. George Guy and his wife Margaret in 1951. Quote, the Guys were determined to grow the first immortal human cells, a continuously dividing line of cells, all descending from one original sample, cells that would constantly replenish themselves and never die. Unquote. But the most important potential application of human stem cells is the generation of cells and tissues that can be used for cell-based therapy. Stem cells can be grown and used more easily than donations. A new artificial skin has already been created using Wharton's jelly mesenchymal stem cells combined with agarose and fibrin, and eventually yields real epithelial cells on the recipient. The need for transplantable tissues and organs far outweighs the available supply. In May 1995, the actor Christopher Reeve was left paralyzed from the neck down after tearing a spinal column in an equestrian competition. He was left with a 20mm gap in his spinal cord. The Man of Steel couldn't even breathe on his own. However, after months of physical therapy and countless procedures before finally regaining feeling below his neck, Reeve toured the country promoting federal funding of stem cell research. What is stem cell research an ethical branch of medicine? There are a few problems people have with it. First is where researchers get the cells. There are 3 types of stem cells. Adult somatic stem cells, which are very few in number and taken directly from a few tissues that are specific for that tissue's regeneration. 2, induced pluripotent stem cells, which are somatic stem cells that are reverted back to their undifferentiated state. And 3, embryonic stem cells, which are taken from embryos grown in eggs fertilized through in vitro fertilization. They're taken from the center of the blastocyst. These stem cells are the main focus of scientific or ethical debate. 2 organizations have taken serious stance against the use of those stem cells are the Illinois Right to Life and the Wisconsin Right to Life organizations. Collecting embryonic stem cells requires killing the embryo, and therein lies the problem because that's committing murder. Secondly, they claim that adult stem cell research should be scrutinized as well because, quote, adult stem cells can also be used to describe cells taken from aborted children. On the other side, many people feel that a human life doesn't truly start until the fetus has a conscience and a beating heart. Until that point, an embryo is just a clump of cells, and as long as there is permission by the donor, stem cells can be collected from the embryo. Proponents ask why the good of the many is jeopardized by the philosophy of the few. The next ethical problem people have with stem cell research is the possibility of human cloning. In cloning, the nucleus of a somatic cell is taken from a donor and transplanted into a host egg, in which the genetic material is removed. Once the DNA is fused, the new cell can begin to grow. This is the process that was successfully used to clone Dolly the sheep. Clones currently have shorter lifespans, usually prone to disease, and have a higher risk of genetic defects. Ethically, this would be setting up a life for failure. Another point opponents make is that our genetic uniqueness is what gives us dignity. Knowing you're exactly the same as someone else would detract from that individual's uniqueness and therefore demolish their dignity. I stand in the firmest affirmation of the use of stem cell research both in embryonic stem cells and the somatic stem cells for 3 main reasons. First, because disease such as cancer are caused by unregulated cell growth, and embryonic cells give us a greater insight to cell division. Secondly, the spare embryos that are used for stem cells are destined to be discarded waste anyway, so why not make good use of them? And finally, the law permits in vitro fertilization, which doesn't consider spare embryos as humans, so why should taking the cells from a discarded one be against the law? Early embryonic stem cell research in the UK has already turned up solutions in the short time it's existed. In one case study, embryonic stem cells were used to repair hearing loss and actually restored 46% of hearing abilities. Is it ethical to own a DNA sequence? The premise of gene patenting, according to the American Medical Association, is a broad term referring to the patenting of genetic sequences such as DNA, RNA, and cDNA. Gene patenting was first done by Amgen in 1990 when the U.S. Patent Office made it a legitimate patent. By 1998, the UK had fully legalized gene patenting. It's done by placing an intellectual property patent on the cDNA, which has had its exons cut out. Pros and cons of gene patenting. First, the pros. It helps small companies compete with larger ones who would otherwise be unable to analyze and process data as fast. It supports exploration by forcing companies to think outside the box, and it provides investment opportunities and makes research profitable. Cons. It hinders research because the owning company doesn't have to pursue every different route. And it will also lead to monopolies on genes and ultimately organisms. It inhibits medical research and results by slowing down the process of testing. And it could put medical care and services financially out of the reach for those who need it most under a monopoly. Most people believe that ownership of DNA and genes is not ethical because it's not exactly ours, nor did we create it. In 2013, the U.S. Supreme Court ruled human DNA cannot be patented because it's a product of nature. However, the court did permit patents on genes created and DNA manipulated in the lab because those were not found in nature. In 1997, a patent for the Canavan gene sequence was issued to 4 researchers in the Miami Children's Hospital. The hospital contacted all labs offering the test, demanding that they stop. Charging more for the test was cruel. Parents who had already donated their children's brains to research had to pay for their tests on other children. As far as the lab-produced genes, most are still against them. They have the potential to monopolize entire industries. It also halts competitive research on genes, reducing the productivity and the amount of possible positive outcomes from analyzing what could be an extremely important gene. However, some support it because it can be used to produce more effective gene therapy drugs. When DNA is used as a chemical compound to produce drugs and vaccines, it's seen as a complex chemical, and a patent makes sense. Recombinant drug production, for example, involves creation of a nucleic acid that did not previously exist in nature, which is then used to produce another medically valuable chemical. Most people just don't understand why the scientific community wouldn't want to share like it always has. For example, Dr. George Guy grew the first immortal line of HeLa cells from the original host Henrietta Lacks in 1951 and spread them around the world willingly. Guy stated that the cells could be used to find a cure for cancer. Quote, and to make that happen, Guy began sending Henrietta's cells to any scientist who might use them for cancer research. Guy sent them via plane, in pockets of pilots, and cargo holds. He sent shipments of HeLa cells to researchers in Texas, India, New York, Amsterdam, and many places between. I stand against gene patenting. There are 3 main reasons I disagree with it. First, it stops progress. It prevents others from working with it. Second, gene patenting adversely affects medical treatment. It's unethical for a company to own a test for a gene. And finally, gene patenting works against capitalism. Without competition, research ultimately will lose purpose and nothing will push its limits and applications. In conclusion, I stand in support of stem cell research because of its applications for regenerative medicine and treating serious injuries and ailments. I do, however, stand in opposition to gene patenting and owning genetic sequences because it adversely affects medical treatment and opposes capitalism. Thank you for your time.